Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome to the deep dive. Today we're plunging into something really fascinating, a breakthrough that kind of flips our usual thinking about gene editing.
Yeah, it's pretty counterintuitive.
Exactly. Imagine using a modified CRISPR system, not to perfectly fix genes, but actually to create, well, targeted, subtle damage.
Damage that specifically takes out cancer cells.
Right, while leaving the healthy ones mostly untouched. That's the core idea we're unpacking today, using a cancer cell's own flaw against it.
It's a really remarkable piece of research, basically turning a precision tool, CRISPR, into a smart bomb for cancer. We're going to explore how scientists use a specific type, Cas9 nickases,
right.
to go after a key vulnerability in lots of cancers, having way too many copies of certain genes, gene amplification.
So our mission today, break down the science, point out those aha moments.
And show you why this could be, a real game changer for future cancer treatments.
Okay, let's set the stage a bit. We all know CRISPR-Cas systems are, well, revolutionary for gene editing. Incredible tools.
Absolutely. Game-changing tech.
But there's been this nagging problem that researchers have kind of wrestled with. These systems often struggle with repetitive DNA sequences.
Yeah, those repeats. They're everywhere in our genome, and maybe even more so in cancer genomes.
And the issue is?
Well, the standard CRISPR tools can get a bit confused by all that repetition. It can lead to less efficient editing, or worse, hitting the wrong spot off-target effects.
Which you definitely want to avoid in therapy.
Exactly. And these repetitive bits, they aren't just junk DNA, they're actually important for how our chromosomes are structured and kept stable.
So targeting them precisely is tricky.
It's been a big technical hurdle. People tried other CRISPR tools before, but they often didn't recognize the repeats well or weren't very efficient. There's been this technical gap.
A need for better tools for these specific regions.
Right. And cracking this could help with more than just cancer. Think genetic diseases, fixing chromosome problems. It's a big deal for genome engineering overall. This new research really offers a very clever solution.
So how do they crack it? You mentioned nickases.
Yeah, the core idea is super smart. They're not using the standard CRISPR-Cas9 that cuts clean through both DNA strands, a double-strand break.
Okay.
Instead, they use this modified version, the Cas9 nickase. It's designed to make only a single-strand break, just a nick.
Like a little scratch on one side of the DNA ladder, not kind of the whole run.
Exactly, a tiny scratch.
And how does that tiny scratch become deadly?
That's the ingenious part. In normal, healthy cells, especially if they're not dividing rapidly, these little nicks, they're mostly harmless. The cell just repairs them, no big deal.
Okay, standard repair mechanisms kick in.
Right. Our cells are pretty good at that.
Yeah.
But in cancer cells that are dividing like crazy and have tons of extra copies of a specific gene, that gene amplification we mentioned,
The vulnerability.
that's where those nicks become a huge problem. When the cancer cell tries to replicate its DNA, which it does constantly, the replication machinery keeps bumping into these nicks.
And all those extra gene copies.
Yes. Imagine hitting the same tiny pothole over and over and over again, but across maybe hundreds or thousands of copies of that gene.
That sounds bad.
It is. Those repeated collisions convert the simple nicks into much more dangerous double-strand breaks. Catastrophic damage, really.
Wow. So it weaponizes the amplification itself.
Yep.
The thing that makes the cancer aggressive becomes its downfall.
Precisely. It's not about fixing the DNA. It's controlled damage triggered by the cancer's own specific genetic flaw.
A true Achilles heel. So only the rapidly dividing cancer cells with those amplified genes get hammered.
While healthy cells, which have fewer copies and maybe divide slower, are largely spared, like you said, a smart bomb.
Okay, so that's the theory. How did they test it?
So they focused on neuroblastoma cells. It's a type of childhood cancer that often has way too many copies of an oncogene called MYCN.
Unknown driver of that cancer.
Exactly. And crucially, they made sure to test cell lines with the MYCN amplification and cell lines without it so they could compare directly.
Good control.
Yes, very important. They designed their tools, the guide RNAs, to lead the nickase right to the MYCN gene. And they included other controls too, like targeting a really common repetitive element called line 1, just to show the nickase could be toxic if it hit lots of targets.
Okay, so they set up a really robust experiment. What happened? Did it work like they thought?
The results were pretty striking. When they targeted that common line 1 element, yeah, the nickase was toxic to all the cells. That confirmed it was active.
As expected, that's the general toxicity check.
Right, but here's the key finding. When they targeted MYCN specifically, the nickase wiped out the neuroblastoma cells that had the MYCN amplification really quickly.
Wow.
And it was dose-dependent. More nickase meant more cancer cells died. But, and this is critical, the cells without the MYCN amplification, or other types of cells, largely unaffected.
That's the selectivity they were hoping for. A clear hit only on the amplified cells.
Exactly. It really shows the power of targeting that amplification.
So that raises a question. Were the cells dying because the nickase was messing up the function of the MYCN gene itself, like stopping it from making its protein?
Excellent question. And maybe surprisingly, the answer seems to be no.
Really?
Yeah, they checked the levels of the NMY protein, the protein made by the MYCN gene, and they didn't see big changes.
Huh.
Which strongly suggests the killing isn't about shutting down the gene's job. It really seems to be about the sheer number of copies, the physical burden of hitting all those copies.
It's the quantity, not the quality, so to speak.
You could put it that way. They even found it didn't really matter where in that amplified region they targeted the nickase. The effect was the same.
Which again points to the copy number being the driver.
Yeah.
Fascinating.
Yeah.
So they saw the cells dying selectively. Did they actually look at the DNA to confirm the type of damage happening? You mentioned nicks turning into double-strand breaks.
They did exactly that. They used a technique called the COMET assay. It lets you visualize DNA damage in single cells. Damaged DNA streams out like a comet's tail under a microscope.
Cool technique.
It is. And they saw a big increase in damaged comet tails in the MYCN-amplified cells when they targeted MYCN, but only minimal damage in the non-amplified cells.
So visual proof.
Visual proof that the nicks were being converted into those lethal double-strand breaks, specifically in the cells with the amplified gene, just like the hypothesis predicted.
Okay, so the damage is confirmed. What were the cells actually doing in response to this attack? Were they just giving up?
Oh no, they were fighting back, or trying to. They saw clear signs of major cellular stress. The cancer cells started piling up in the S phase of the cell cycle.
That's the DNA copying phase.
Right, and then they got stuck in the next phase, G2M, unable to divide properly. It indicates severe replication stress. The cell hits the brakes, trying to fix the damage.
Alarm bells ringing inside the cell.
Totally. They found molecular markers of this stress, like higher calcium levels, more reactive oxygen species, basically cellular panic signals.
Yeah.
And the cell's own DNA repair systems, particularly one called the ATR-CHK1 pathway, went into overdrive.
Trying to patch things up.
Desperately. They even did some clever experiments where they messed with certain repair enzymes. If they reduced one called XO1 or boosted a protective protein called RPA, the cells survived a bit better.
Which gives clues about the repair pathways involved.
Exactly. And maybe ways to make the treatment even more effective later on, perhaps by blocking those repair attempts.
So the cells try to cope, they get stressed, they try to repair, but ultimately it's too much for the amplified cells. How do they finally die? You said it wasn't typical apoptosis.
Right. It wasn't that neat programmed cell death. It was messier. They saw signs of something called mitotic catastrophe. The cells basically try to divide even with all that unrepaired DNA damage.
And that doesn't end well.
No. It leads to a form of necrosis cell rupture. This was driven by an enzyme called PRP1 going into hyperdrive, which burns through the cell's energy.
And the cell basically runs out of fuel and falls apart.
Pretty much. And importantly, when they use drugs that inhibit PLP1 or another enzyme involved called calpain, they could actually rescue the cells significantly.
Which pins down the specific death pathway. Very cool.
It really maps out the whole process from the initial nick to the final cell death.
This whole approach, it really feels different, like a genuine shift.
I think it is. When you step back and look at the bigger picture, you see how it tackles some major unmet needs.
Okay, like what?
Well, first, that technical gap we talked about, CRISPR struggling with repeats and amplifications. This nickase approach nails it. And these amplifications are common, maybe over half of cancers, especially the more advanced ones.
So it opens up a whole new set of targets.
Exactly. Second, The mechanism is new. It's not blunt cutting like standard CRISPR. It's subtle nicks, weaponizing the cell's own replication process. Less risk of causing random, unwanted mutations.
That leads to safety, right, which is always huge.
Absolutely. That's the third big point. The selectivity and safety look really promising. Compared to standard Cas9 cutters, this nickase showed way fewer off-target hits.
Hitting only what it's supposed to hit.
Mostly, yeah. And critically, it caused minimal damage in healthy, non-amplified cells. They even tested sensitive cells like blood stem cells and non-dividing nerve cells.
And they were okay.
Largely, yes. Which significantly lowers the risk of side effects, a major hurdle for any gene therapy. It just seems inherently safer because it relies on that cancer-specific vulnerability, the amplification.
Safer, precise, new mechanism. But is it just for neuroblastoma or MYCN?
Great question. And that's the fourth point, its potential breadth. They didn't stop with neuroblastoma or MYCN.
Oh, good.
They showed it works in other cancer types too, targeting different amplified genes like ERBB2 or HER2 in breast cancer.
A very well-known target.
Right. And MYC in lung and colorectal cancers. So it seems broadly applicable.
That really strengthens the case.
Definitely. And they even showed you can boost the effect. Combining the nickase with drugs that block DNA damage repair like CHK1 inhibitors made it kill cancer cells even better. Synergy.
Using existing drugs to enhance the new approach.
Exactly. And they even showed hitting 2 different amplified genes in the same cell at once worked even better. Multiplexing.
Wow, so it's potentially quite versatile.
It really seems to be.
So let's tie this all together. What's the big take-home message here? How does this change things?
I think the big picture is we can potentially take a gene editing tool known for precision, the nickase, and use it not for editing, but as a highly targeted therapeutic, one that cleverly exploits a cancer's own genomic instability, those extra gene copies against it.
Turning the cancer's strength, in a way, into its fatal weakness.
Perfectly put. It opens up possibilities for really specific, maybe even personalized cancer treatments.
Yeah.
Moving beyond just blasting all dividing cells. But, there's always a but in science.
The next challenge.
The next big challenge. This all looks fantastic in cell cultures in the lab. The crucial next step is figuring out delivery. How do you get these nickases into the tumor cells efficiently and safely inside a patient, in vivo?
Right. Delivery is always the key hurdle for these kinds of advanced therapies.
It is. Solving that will be essential to translate this really powerful discovery into something that actually helps patients.
Well, that's our deep dive for today. It is genuinely mind-blowing, the idea that just a tiny nick in DNA can trigger a death sentence for cancer cells, leveraging the very chaos that defines them.
It's just brilliant science. Really shows how understanding the absolute fundamentals of cell biology can lead to completely new ways of thinking about treatment, often unexpected ways.
So as you mull this over, here's a thought to leave you with. If we can use a cancer cell's extra DNA copies to destroy it, what other subtle genomic quirks, things beyond just amplification, might be hiding other vulnerabilities?
Yeah, what other weaknesses are lurking in the messy genomes of cancer or other diseases?
Just waiting for an equally clever, maybe equally counterintuitive solution.
Lots to think about.
Thank you for joining us on the Deep Dive. We hope this look at turning CRISPR nicks into cancer killers leaves you informed, maybe a little amazed, and curious to keep exploring. See you next time.